Hey everyone, and welcome to RUBIN'S Mind Lab, where we break down the latest research into what it really means. Ever wondered if we can figure out if you are more like your twin brother or sister genetically, even if nobody knows for sure if you are identical or just regular twins? Well, this paper dives deep into that tricky question. So basically, scientists love using twins to study nature versus nurture. They compare identical twins who share almost all their genes with fraternal twins who share about half their genes, like regular siblings. By seeing how similar twins are in a certain trait, they can guess how much of that trait comes from genes or from the environment. But here is the catch. You absolutely need to know if the twins are identical or fraternal, meaning you need their zygosity information. If you don't have that, it is like trying to solve a puzzle with missing pieces. This paper looks at a clever method called the normal finite-mixture-distribution twin model. Think of it like this. Instead of knowing for sure if a twin pair is identical or fraternal, this model tries to figure it out from the data itself. It assumes that identical twins and fraternal twins will look a bit different in their outcomes. So it can try to split the total group of twins into these two possible subgroups. It is a really cool idea because it could let us study genetics in huge groups of twins where we don't have that important zygosity information. This is great for big population studies or when you are looking at data from educational platforms like the one they use. But here is where the fun and the problems start. This model has a big assumption. It assumes that within each group, the identical twins and the fraternal twins, their traits are spread out normally. You know, that bell curve shape. The tricky part is, since you don't know if they're identical or fraternal, you can't actually check if this assumption is true for each group. You can only see the combined group. So, the researchers did a huge simulation study. They basically created tons of fake twin data. They played around with different factors like sample size, how much of the trait was due to genes versus environment. And importantly, they messed with the normality assumption. They wanted to see how much trouble the model got into when this normality assumption was broken. Did it give wrong answers for how much was genes versus environment? They also tried a trick to fix the problem. Sometimes data isn't normally distributed because it is a bit skewed or has fat tails, things that make the bell curve look a bit wonky. They tried changing the scores a little bit to make them look more like a normal bell curve before running the model. This is called a transformation. They wanted to see if smoothing out the data this way would help the model give better answers. After all the computer simulations, they took this model for a spin on real data. They looked at kids playing educational math games on a platform called Math Garden. Since they didn't know the zygosity of the twin pairs in their data, they used this normal finite mixture distribution model to try and figure out the genetic and environmental influences on math skills and how much kids engaged with the platform. Now, one of the ideas about this model was that you could look at the difference in scores between the twins. If the differences looked normal, it was a good sign the main assumption was okay. But if the differences were weird, like too many extreme scores, that could be a red flag that the normality assumption was violated. The researchers really dug into this idea, trying to see if looking at these differences could reliably tell them if the model was giving a bad answer. But they found a really interesting problem with using the twin differences as a diagnostic. Because you don't know zygosity, the difference scores are a mix of identical twin differences and fraternal twin differences. And even if the scores within each group are perfectly normal, the combined mix of differences can look not normal. Specifically, it can look leptocortic, which just means it has more extreme values or fatter tails than a normal distribution. This happens naturally if there are any genetic influences at all. So, seeing a not normal difference distribution doesn't automatically mean the model's core assumption is broken. It could just be normal twin data showing its twinness due to genes. This makes it super hard to tell if the non-normality is a real problem for the model or just a normal part of twin resemblance. And here is the big finding that shook things up. They discovered that when the data really wasn't normal in a way that the model didn't expect, especially when there was extra kurtosis or those fatter tails in the twin difference scores, that wasn't just due to the A structure. It really messed up the results. The model gave seriously wrong answers about how much was genes and how much was environment. Remember that trick of transforming the scores to make them look more normal? It turned out that didn't really help much. It didn't make the model's wrong answers any better. So that potential fix wasn't a magic bullet. They also tested a bivariate version of the model. This means looking at two traits at the same time instead of just one. The cool thing here is that if one trait has weird non-normal data, but the other one is nicely normal, the model might be able to use the normal one to help correct or stabilize the estimates for the weird one. And they found that this bivariate approach did help a bit. The wrong answers weren't as bad as when they looked at just one trait alone, but the problem wasn't completely solved. Now, for the real-world data from the math games, the univariate models, looking at one math skill at a time, gave really wild results. They said that things like addition, subtraction, multiplication, and even how often kids logged in were like 91 to 99% heritable. That is almost entirely genetic, which is super high and frankly not very realistic for things like math skills or logging into a game. For comparison, in other studies, math skills are usually found to be around 60 to 74% heritable. The only exception was division, which had a more reasonable heritability. When they used the bivariate models, the heritability estimates weren't as ridiculously high. They were still high, but a bit more toned down. And importantly, they found more environmental influence, especially shared environmental influence, which they expected for things like logging into a game. However, the results from the bivariate models jumped around a lot, depending on which traits they paired together, making them unstable. So, while they were less biased, they weren't very reliable. The big takeaway from this research is that while the idea of a mixture model for twin analysis without zygosity is promising, it is also really tricky. If the data doesn't follow the model's assumption of normality within each twin group, it can lead to seriously biased results. And it is hard to even spot that bias because the usual checks aren't always reliable. The bivariate model helps a bit, but it doesn't completely fix the problem. So their conclusion is pretty strong. For now, they don't recommend using this model in its current univariate or bivariate forms for making firm conclusions about genetic and environmental influences. It is not that the idea is bad. It is just that the model isn't robust enough to handle real-world data that often deviates from perfect normality. This is a bummer because the goal was to unlock genetic research in more diverse and larger datasets. Looking back at the simulations, they found that the model worked best when the sample size was really large, over 2,000 twin pairs, and when the heritability was already quite high. This means it might work okay in very specific, ideal situations, but in more typical research scenarios, especially with smaller sample sizes, the risks of bias are substantial. This part really highlights the core issue. The normal finite mixture model relies on the idea that if the twin differences look normal, then the underlying assumptions are probably met. But as they showed, the twin differences can look not normal, even when the data is fine, just because of the genetic relatedness. This means you can't trust the normality of the twin differences as a foolproof sign that everything is okay. It is a diagnostic that is not very good at diagnosing the specific problem it is supposed to detect. This is a really important nuance. When the twin difference scores are negatively kurtotic, meaning they have fewer extreme values and more moderate ones than a normal distribution, that is a big red flag. The normal ACE model, even with its inherent differences between MZ and DZ twins, tends to produce either normal or positively kurtotic distributions. So, when you see negative kurtosis in the differences, it strongly suggests something else is going on, some other source of non-normality that is likely causing bias. So, what is next? The researchers point out that while transforming the data didn't help this specific model, maybe other transformations could work better. They also suggest that future research could explore even more complex multivariate models, looking at many traits at once. The idea is that if you have several traits and one of them is a bit wonky in terms of normality, the other, well, behaved traits might help balance things out and lead to more reliable overall estimates. It is a promising avenue, but it needs more testing. This is an interesting point about data sharing. The data for the math game study is actually owned by the company that developed the platform. This means it is not publicly available, which is a common issue in big data research. They've made their code available, and a preprint of the paper is online, which is great, but accessing the raw data would require special agreements. Ultimately, this study is a really important caution. The normal finite mixture distribution twin model is a clever idea for dealing with missing zygosity information, and it has a lot of theoretical appeal for unlocking insights from large datasets. However, this research makes it clear that in its current form, it is too easily fooled by normal data. The bias it can introduce is significant, hard to detect, and can lead us to believe things about genetics and environment that aren't really true. When they looked at the real data, the results were pretty extreme, and the additional checks like comparing twin correlations to classmate correlations helped highlight these issues. The fact that the twin correlations in the math data weren't as high as the model predicted, given its own very high heritability estimates, was another clue that something was off. It is like the model was saying genes, 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 while the actual twin similarities were saying maybe not that much. It is fascinating to hear the researchers admit that their own findings are contrary to what they had hoped for. They started this research with the goal of validating and refining a promising method, but the evidence led them to a more critical conclusion. This shows the integrity of the scientific process. They weren't afraid to reveal that a method, despite its good intentions, might not be ready for prime time yet. And while they're cautioning against using this specific model now, they're not giving up on the overall goal. They emphasize that finding ways to do genetic research on large datasets without knowing zygosity is still a really important area for future work. This study has provided crucial insights into why it is so difficult and what pitfalls to avoid moving forward. It is a call for more sophisticated models and better ways to handle real-world data imperfections. So, what is my take on all this? This study is a fantastic example of rigorous scientific investigation. The researchers didn't shy away from testing a popular method under challenging conditions. They used simulations to meticulously understand the model's behavior and then applied it to real data to see how it held up. Their honest conclusion that the current model is too prone to bias for reliable real-world use is actually incredibly valuable. It prevents other researchers from potentially drawing flawed conclusions. It highlights the critical importance of data assumptions in statistics. They've shown us that just because a method seems clever doesn't mean it is always accurate, especially when assumptions get bent in messy real-world data. For future research, it is clear we need models that are more robust to non-normality, or at least provide clearer diagnostics of when that non-normality is a problem. Maybe adding more traits or using different statistical approaches will be key. It's a reminder that in science, sometimes the most valuable findings are the ones that tell us what doesn't work, guiding us toward better solutions. What do you all think? Does this surprise you? Let me know in the comments below. And if you enjoyed this deep dive, please hit that like button, subscribe to RUBIN'S Mind Lab for more explorations into the fascinating world of psychology and the human mind, and ring that notification bell so you don't miss our next video. Thanks for watching.